Hello. Hey, good morning, Nishita. How are you? Am I audible? Okay, Nishita, can you tell me a little bit about yourself? Hello. Can you hear me?
Yes, I hear you.
Yeah. So can you tell me a little bit about yourself? Anything.
My name is Nishchita. I'm studying in 9th grade in Navodaya school,
Okay.
and it's a CBSE school.
All
And
right.
I am from Kolar.
All right. Okay. All right, Nishita. So tell me what's happening in school, Nishita. Like what chapter has been going on? Or any
The
particular
the cell chapter is completed.
cell is completed. Okay, great. All right. So any particular topic that you want to do today, or you want me to pick up the topic for demo?
You can just revise the chapter once.
Just give me a moment. I'm just sharing my whiteboard, okay? Okay, can you see my screen?
Yes, I can see.
All right. Let's talk about cell now. So, can you tell me how do you define a cell?
Cell is a basic and fundamental and a structural unit of life.
Correct. So, cell is the structural and fundamental unit of life, right? So, what does structural and fundamental unit of life mean is that structure wise, also, this is the most basic or the smallest unit of life. That means you cannot divide the organism lesser than a cell, right? It is the smallest unit structurally and fundamentally, functional wise also, a cell can perform all the basic functions that a multicellular organism can perform. So, for example, let's say we do our body is undergoing digestion, reproduction, respiration, excretion. All these uh, metabolic activities can be done by a single cell also. That is why it's also known as the fundamental unit of life. That means functionally also it's the smallest unit. Okay. Excuse me. All right. Now, sorry. All right. So cells can be divided into different types based upon their organization internally and the organization of one particular organism. So cells can be classified as prokaryotic cells or eukaryotic cells. Can you quickly tell me the difference between prokaryotic and eukaryotic cell?
Prokaryotic means it's nothing but a primitive cell. It
Correct.
is not so developed. It does
Correct.
not have much of the cell organelles which is included in the eukaryotic. It
Correct.
it can be seen in blue green algae,
Hmm.
and it was mostly present in the early times.
Correct.
And eukaryotic cells are mostly available in all the types of organisms, and
Correct.
they have a lot of cell organelles, and they have different organelles for every different functions.
Correct. So pro means, like you said, it's primitive, right? So pro means primitive. And karyon or karyotic. Karyon means nucleus. So prokaryotic means primitive nucleus, like you said. True, U means true. And karyon again means nucleus. So eukaryotic means they have a true nucleus with the nuclear membrane and everything. All right. So prokaryotic organisms are always unicellular. Okay. They have no nuclear membrane. That is why a primitive nucleus. And they have no membrane bound organelles. Okay. Examples of prokaryotes, we have all the archaea and bacteria. Right. Coming to eukaryotic organisms have a true nucleus, so they will have they can be both multicellular or unicellular in nature. All right. One second. They have a well-defined nucleus. And they have all the membrane-bound organelles are present. Okay. Examples. If I talk about a unicellular eukaryotic organism, we have yeast, amoeba, okay? And multicellular, we have plants and animals. Right? Any doubt so far here? Okay.
No.
All right. So like I said, when we are talking about cells are divided on the basis of one, their internal cellular organization. That means how they are internally placed, for example, prokaryotic, eukaryotic. Another distinguishing characteristic is the number of cells, which is whether they're multicellular or unicellular. All right. So unicellular, we've all seen that prokaryotes are always unicellular. Eukaryotes can be unicellular also and multicellular also. All right. Now I'll give you an overview of how a cell is organized, and then we'll first talk about a prokaryotic cell in detail. So a cell can be divided into cell envelope, okay, and the protoplasm. Couple of terminologies are normally used interchangeably, specifically in NCERT, right? That is a problem with NCERT. They sometimes use protoplasm, they sometimes use cytoplasm, then they'll use cytosol, but you need to know the difference between all of them. So cell envelope can be of two types, of course. We have the cell wall and we have the cell membrane or the plasma membrane. Okay. Protoplasm, on the other hand, is cytoplasm plus the nucleus. Okay, so anything that is present inside the cell membrane is protoplasm. Protoplasm minus the nucleus is the cytoplasm. And cytoplasm is made up of cytosol plus all your other organelles. Okay, and these organelles will include mitochondria, Golgi bodies, endoplasmic reticulum, ribosomes, plastids if we are talking about plants. Okay. So many such organelles are present, which we'll discuss in detail. So any doubt between these terminologies, protoplasm, cytoplasm, and cytosol?
No, ma'am.
Okay. Shall we now look at the prokaryotic cell first? So prokaryotic cell we know is a primitive cell. And all your bacterial cells are prokaryotic. So how does a prokaryotic cell look like? Is first it will have a outer covering, which is the cell wall, right? Inside the cell wall, you will have the cell membrane, and the nuclear material is present in the cytoplasm like this only. And this region where the DNA material is present is known as the nucleoid region. So you have your cell wall, you have the cell membrane. Okay, this is the DNA present in the nucleoid region, right? Other than that, you will have ribosomes everywhere. Ribosomes are membraneless organs, so they are present in the prokaryotic cell. Okay. Apart from this, they can have a flagella for locomotion, or their body can be covered by these hair-like structures known as fimbriae. Okay. And whatever is remaining is your cytosol. Any doubt here?
No, ma'am.
Okay, now let's talk about each and every part in detail. So first we have the cell wall. So cell wall and cell membrane form the envelope of the cell. Cell wall in bacteria is made up of peptidoglycan. Peptidoglycan means, peptido means protein and glycan means glucose or sugars or carbohydrates. So it's a mixture of both proteins and carbohydrates. Okay. Cell membrane is same as all eukaryotic cells. Cell membrane is universal, so it's a phospholipid layer. This we'll discuss in detail. So phospholipid bilayer and proteins. Okay. DNA here is circular. Okay. Not number of chromosomes like we have or eukaryotes have. They have a single circular DNA. Nucleoid is the region where DNA is present. Ribosome is 70S type. Okay. Cytosol will have some enzymes. Fimbriae helps in locomotion. It helps in conjugation. It helps in capturing food. And flagella will help in locomotion. Okay. Now, do you know what is a 70S ribosome?
No, ma'am.
Okay, so what happens is there are two types of ribosomes, okay? Prokaryotes have 70S, eukaryotes have 80S. Ribosomes are made up of two subunits, right? There's a large subunit and there's a small subunit. And they will come together when protein synthesis needs to occur. So 70S, S is nothing but a unit that we use to measure the amount of proteins that is present. So S is your Switzberg unit, named after a scientist. This unit measures how much proteins are present. So in 70S ribosome, in the large subunit, we have 50S amount of proteins, and this is 30S. This is your 70S ribosome, only found in prokaryotes and certain organelles inside the eukaryotes, like mitochondria and plastid. Okay. Whereas in eukaryotes, you will have a larger ribosome. So 60S will be large subunit, 40S will be small subunit, and this is an 80S ribosome. Okay. So for now, you don't need to know the details of the kind of proteins that are present inside. You just need to know the difference, that one is smaller, 70S, one is larger, 80S. Okay, so prokaryotes have a smaller ribosome. Any doubt?
No, ma'am.
All right, so this is your prokaryotic cell. When I talk about each and every organelle in detail, whatever is the difference, I will still mention it there so that in the end, you can make a table of prokaryotic, eukaryotic, plant and animal cell like that. Okay. All right, now let's talk about the cell envelope. Oh, one more thing I wanted to tell you here. Yes, sometimes what happens is, this is good. Sometimes there is an additional covering on the top of the bacteria. This is known as a capsule or slime mold, okay, or a slime layer. This is an additional protective layer that is present on some of the species of bacteria. Okay, this layer will make it more virulent. Why? Because whenever we get a bacterial disease, what we do is we take an antibiotic, right? So what does the antibiotic do? Antibiotic will go and penetrate the cell wall and will not allow the cell to reproduce. Okay, so it breaks the cell wall. So automatically the cell also breaks down. But if there is an additional protective covering on top of the cell wall, it's very difficult for the antibiotic to break through. And that is why this one is more virulent. Okay. All right. Now coming to the cell envelope. So one by one, we will talk about each and every organelle. So cell envelope, first we'll talk about the cell wall. So cell wall is a dead component of the cell. Okay. So anything that is inside the cell membrane is living. So if a cell has a cell membrane, then we call it a living cell. Otherwise, if only cell wall is present, like in the case of cork cells or bark cells of plants, they are dead. So only cell wall is present, outer portion. Cell wall is present in plants, bacteria, and fungi. Okay. In plants, cell wall is made up of cellulose. In bacteria, it is made up of, I've already told you, peptidoglycan. And in fungi, it is made up of chitin. All of them are derivatives of carbohydrates. All right. So cellulose is also a complex.
Can you repeat what the fungi is made up of?
Chitin. 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 So that is also a complex carbohydrate. CHITIN. Okay. CHITIN. So this is also a complex carbohydrate like cellulose, and peptidoglycan is also a derivative of glucose only. Glycan is glucose and peptido is protein. So it's a dead component mainly for providing rigidity, protection, and transport. Cell wall is a freely permeable membrane. That means it allows all substances to pass through it. Since it's a dead component, right? So it doesn't really stop anything from going inside the cell or coming out of the cell. That's the job of the cell membrane, right? Any doubt here in cell wall?
No, ma'am.
Okay. Now let's look at plasma membrane or the cell membrane. So plasma membrane is made up of, it has different components. It has lipids, proteins, carbohydrates, and sterols. Okay, so lipid is a molecule that is hydrophobic in nature. So there are two layers of lipids. Okay, so if you look at a lipid molecule, what a lipid molecule has is a lipid molecule is made up of a head, which is made up of glycerol, and it has two chains which are made up of fatty acids. So this head is made up of alcohol, specifically glycerol, and the tails are made up of fatty acid. Okay, now alcohols are miscible in water. If you take alcohol in water, they'll mix, right? Alcohol is soluble in water. That is why the head is hydrophilic in nature. That means hydro means water, philic means loving. So this is water loving. Fatty acid, on the other hand, oils and all fats, they are made up of, they are hydrophobic. They don't mix with water easily, right? So the tails are hydrophobic. Phobic means they are scared of water. They'll not mix in water. So the lipid bilayer is arranged in this manner. The heads are towards the outside and the tails are towards the inside. This is how the lipid bilayer is arranged. This is a very basic model that I'm showing you. Of course, we'll do the diagram also in detail, right, when we come to the regular class, but this is just for explanation. So this is your lipid layer. Since lipid layer, lipids have phosphate groups attached to it. That is why they are known as phospholipids. So that is why we call them phospholipid bilayer. Okay. Now there are proteins present. So there are different kinds of proteins. Either they are spanning the entire membrane or they're only present on the periphery or only on the corners. The ones that are spanning it are usually called as transmembrane proteins or intrinsic proteins. Intrinsic because they are going inside. All right. And the ones that are at the periphery are known as peripheral proteins or extrinsic proteins. They are outside only. So mainly proteins are helping in transport. Okay, either they will form tunnels or they'll form gates or they'll simply attach one molecule and leave it inside and come. This we'll discuss in detail when we come for regular classes. Okay, I'm just giving you an overview right now. So since the lipid bilayer is unable to interact with a lot of molecules, the plasma membrane is a selectively permeable membrane or a semi-permeable membrane. Okay, because most molecules can't interact with the fatty acid chains. So the proteins will help. So proteins either form tunnels, like, an opening where through and through it will pass through, or they'll form gates, which will open and close depending upon the amount of substance to be entered. So that we'll discuss in detail later. Then we have carbohydrates. Carbohydrates will also help in transportation. There are certain molecules that can easily interact with carbohydrates because carbohydrates are simply made up of carbon, hydrogen, and oxygen. And most of your macromolecules or biomolecules that are present are made up of calcium, hydrogen, and oxygen, right? So they can easily interact with the biomolecule or the carbohydrate. Okay, next, normally carbohydrates can be attached in two ways. I'll just draw it with another color. So carbohydrates either can be attached to the lipid layer or they can be attached to the protein. Okay. If they're attached to the lipid, they are known as glycolipids. Glyco means glucose, lipids because they're attached to the lipid. And if they're attached to the protein, they are known as glycoproteins. Okay. Other than that, we also have sterols. Sterols are present in the membrane for strengthening the membrane. Okay. So it will provide a little bit of strength. In animals, cholesterol is present. And in plants, ergosterol is present. So this is for animals. And this is for plants. Okay, this will simply provide a little bit of strength to the cell membrane. So you must have heard that, if you have a lot of cholesterol in the body or you eat a lot of fats, the cholesterol goes and clogs the arteries or the hearts. It's because of that only, because it's already present in the plasma membrane. So if it's in excess, it will get stored in the plasma membrane more and more, and the walls will become thicker. The moment the walls become thicker, arteries have a chance of getting blocked. Okay. So, since plants don't have anything called a cholesterol, it's something called an ergosterol. Okay, so this is your structure of plasma membrane. This model of the structure of plasma membrane is known as the fluid mosaic model. Excuse me. Given by Nicholson and Singer. These are the two scientists. So why a fluid mosaic model? Because the fats or the fatty acids are fluid in nature. They are slightly flexible, and these substances, glycoproteins, glycolipids, proteins, cholesterol, they're all embedded in the layer. Hence, it's a fluid mosaic model. Okay. Any doubt here?
No.
Okay, so I'm going to stop here. In the regular class, we'll talk about the cell theory and all, a little bit about the history and everything. That's also important. Any questions for me, Nishita? Do you have any questions that you would like to ask me or anything?
No, ma'am.
No. Okay, all right, then I'll end the demo here. You can get in touch with the admin, whoever you've spoken to, and we can fix the days and whatever. All right?
Yes, ma'am.
Okay, then I'll see you. Bye-bye. Thank you for joining.
Thank you, ma'am.
Welcome. Bye.